Hello, good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Ernest Atto from Business Review Webinars, and I will be your host. It is our pleasure to have Capsugel with us today, who will be discussing on the webinar entitled Novel HPMC Capsules, Bioequivalence, and Functional Performance. Today's guest speakers are Matt Richardson, a manager at Pharmaceutical Business Development at Capsugel, and Michael Morgan, a senior principal scientist at Capsugel. I would like to welcome you to our webinar platform on 24. This webinar is browser-based, so if you disconnect, click the link you received via email to rejoin the session. All of the icons along the bottom of your screen are the interactive widgets that we have on offer for you today. So please interact with them all throughout the session. You can ask questions using the Q&A widget or by using the questions box. We will try and get through as many of your questions as we can once the presentation has concluded. Please use the help widget if you experience any technical difficulties or require any more assistance. Without further ado, please allow me to welcome Michael and Matt. Matt, over to you now.
Thank you, Ernest. And I'd like to thank the members of the audience for their participation in today's webinar. So today, Mike and I are hoping to demonstrate the following ideas to you. That two-piece capsules made from HPMC have been successfully used as an alternative to gelatin, and you'll see us use the term HGC, or for hard gelatin capsule, throughout the presentation. That these new HPMC polymers have actually eliminated the use of secondary gelling agents, which were so predominant in the first generation of HPMC. And that's going to give us a much more gelatin-like performance that we'll see. We're going to take a look at these next generation of capsules as how we've gone to the next level and made them into application-type capsules as well. And while we do this, we want to talk about the in-vivo performance and compare that from gelatin to HPMC for anybody that's considering new projects or folks that are considering switching over. There are going to be certain circumstances where the API and formulation are going to be equivalent, and we're going to discuss those opportunities with you today. And also, lastly, but as importantly, we want to take a look at some of the investigation work that's going on today on how the HPMC capsule can inhibit drug crystallization from supersaturated states. And then following the webinar, we'd love to address your questions about the materials that are presented here as well. So in the first section of the webinar, we want to discuss the properties, physical and chemical, of the next generation HPMC capsule. And to do this, I want to use gelatin as a baseline of comparison because it's been the industrial standard for so many years. Gelatin raw material is produced from the hydrolysis of collagen, derived from bones and skins of both beef and pork. Those special blends of fish are available. Gelatin is ultimately a mixture of amino acids, so it needs no additive to gel on its own, that occurs at a particular concentration when the temperature of the solution reaches below 40. So we'll melt the gelatin, and as it cools, it will start coming out of solution. So we're going to make a hot solution of the gelatin and dip cooled stainless steel mold pens into the liquid gelatin, and that deposits gelatin onto the pen. As gelatin comes out of the solution, it forms a 3D chain network that you can see on the right side of the slide. And after a drying process, the capsule's got about 12 to 15% water content. The water content is important for gelatin in order to stay nice and flexible. It acts as a functional plasticizer for the hard gelatin capsule. And when the water content goes out of that range significantly, it's got an effect on the performance of gelatin. HPMC is a very different material altogether, though. Cellulose is extracted from white pine trees and then substituted appropriately by the chemical reactions that you see on the slide. We obtained this raw material to process into capsules at several of our hard capsule manufacturing plants in a couple of different manufacturing processes. So the first generation HPMC capsules came into the market, I'd say roughly 15 to 20 years ago. And they were made in a similar process as the one I described above for gelatin. So there's a heated polymer water solution is made. Cooled stainless steel pens are dipped in it. However, in order to get a significant amount of HPMC into solution, into a hot solution, and deposited into the mold bar pens, gelling systems were required. So as customers globally tested that first generation HPMC capsule, they experienced variability in vitro with the capsules, and they relayed those concerns back to us. And they also expressed some interest in capsules that could demonstrate less variability. As the variability was really attributed after study to the addition of gelling agent and gel promoter, it was clear that an alternative process was needed in which gelling systems were not used at all. So based on their feedback, we went back to the engineering drawing board, so to speak, and looked for a way to manufacture HPMC capsules without a gelling agent. In order to do this, we took advantage of the solubility of HPMC, which was actually higher in cold water than in hot water, and that explains why gelling agents were needed in the first generation process where hot water was used. But having a cold concentrated solution of HPMC called for us to use heated stainless steel pens to deposit the polymer onto. And so the engineering and manufacture of the second generation HPMC system actually calls for a completely reversed and new process to manufacture them. So let's take a look at the performance of these capsules, the second generation, which are made by the thermo-gelation process and contain no gelling systems within them. So one major aspect to consider is what I call that window of opportunity, or the window of success with respect to moisture. Gelatin capsules have always been very robust capsules for a multitude of purposes, and we recognize and we've always advised that they be kept within a recommended storage window, and that is 35 to 65% relative humidity, to ensure that the gelatin remains at that 12 to 16% water content for optimal performance. Straying too far below that level, we start to see brittleness begin to come into play, and too far above that level leads to manufacturing issues when the capsules soften and lose shape. As the thermo-gelation, or what we'll call Vcaps Plus capsule by our trade name, are made of HPMC, it doesn't rely on the water content for pliability as gelatin would. And so that window of opportunity is actually larger than what you would find of gelatin. And you see that in the graph on the right side. So HPMC capsules can go as low as 2.5% to 3% before we start to see capsule brittleness introduced. And even then, a very low level of brittleness is introduced. So in early studies, though, for most formulators, how hygroscopic or moisture sensitive a molecule really is may not always be well defined. So starting with a capsule that better levels that playing field is an excellent idea. And it just removes the concern of capsule brittleness and having less concern when desiccants, things like that, are used for early successes. So as importantly, HPMC capsules contain 2 to 9% water content, and that's an average that you'll see 6% less water content than gelatin capsules. And that remains consistent over that lower moisture range, higher moisture range across the field. You can see the differences in the moisture levels pretty clearly when you compare the isotherms both of hard gelatin capsules, that's what we have noted in blue on the graph, versus that of the thermo-gelation HPMC, the Vcaps Plus capsule, which is noted in the red. The graph we show here shows how robust that second generation HPMC capsules, how robust they are, even at low humidities, well past what those are recommended or tolerated for gelatin. So capsules were equilibrated to various relative humidities inside a specific environment. And internally, we use a device called the tube tester that you can see on the left-hand side, which drops a 100-gram weight on the capsule. And then we would inspect the capsule for any sign of dent, crack, or breakage. So we'll take typically a 100 capsule sample, expose them to the different relative humidities, and then test them for breakage. And what you see is while gelatin begins to lose its water, which again acts as its plasticizer, the capsules are more prone to and begin to show signs of breakage, which increase with the loss of moisture. Compare that in orange, though, to the HPMC capsule, which do not show anywhere near the same level of adverse effects at lower moisture levels than the hard gelatin capsules do. And this makes the HPMC capsule very suitable for low moisture applications, hygroscopic compounds, use of desiccants, and similar work. We can see the impact of this as well with the very moisture-sensitive compound acetylsalicylic acid. So in this experiment, we took the aspirin formulation and it's placed in both HPMC and gelatin hard capsules on two stability platforms. The first at 25 degrees Celsius, 60% relative humidity, and the second at 40 degrees centigrade and 75% relative humidity. The capsules were placed in inductively sealed HCP bottles so that environmental concerns were negligible, and the amount of degradation of the aspirin was measured over time. In both cases, it can be seen the amount of degradation is actually higher in the gelatin capsule shell as compared to the HPMC capsule shell. So after 18 months at 25 degrees and 60% relative humidity, there is approximately 8% degradation in the formulation contained in the hard gelatin capsule, noted in blue, as opposed to the 2% in the HPMC hard capsule that you'll note in orange. Similarly, at accelerated stability, the same effect can be seen both at 3 and at 6 month time points, with approximately 5% of degradation of the formulation in the hard gelatin capsule can be found as opposed to just over 1% in the HPMC capsule. And again, we attribute this to the moisture difference between the gelatin and the HPMC capsules. As the capsules are inductively sealed, we can effectively rule out any contribution from the environment. So the moisture in the shell itself has to be responsible for any product degradation. As we saw on the previous slide, HPMC is 6% median water content as compared to gelatin's 14%. And this would explain the differential in degradation. Okay, so also looking at some of the physical characteristics, the thermo-gelation, the second generation HPMC capsule, without a gelling system in it, shows better weight consistency than capsules that contain, HPMC capsules that do contain the gelling agent. So in this review conducted by Koo and others, the Vcaps Plus had 3.29 relative standard deviation, compared with first-generation capsules that do contain gelling systems having a higher 6.45 relative standard deviation. And similarly, differences in the powder leakage can be looked at. If we examine the capsules by scanning electron microscopy of the shell surfaces, we look across the 3 different types, and we've got those presented here. So the thermo-gelation capsule, the Vcaps Plus, which we'll designate as shell 2, has a very clean edge, very smooth surface compared to the other 2 capsules. Either hard gelatin, which is denoted by HGC, or shell 1, which represents the traditional HPMC capsule containing a gelling system. The maximum gap between the body and the cap, as well as the measurement of the shell thickness at the closure cross-section, is really summarized in the photo below. The scanning electron photomicrographs show that hard gelatin has the thickest shell, followed by the hypermellose, the thermo-gelation shell, the Vcaps Plus, and then in third, the hypermellose shell 1. The shell thickness variations are very similar. The shell 1, which contains the gelling agent, has a large gap between the body and the cap, twice as much as the gap that you'll find for the hard gelatin capsule. And then the shell 2, containing no gelling agent, has a slightly larger gap than gelatin, but significantly smaller than the hypermellose shell 1. The researcher wrote that an improvement in the powder leakage quality attribute can be expected from the HPMC capsule because that gap is reduced significantly. And indeed, when tested in blister carding and seal integrity, we found no powder leakage was observed in the replacement of hypermellose shell 1 by the hypermellose shell 2. So when switching over, we see a drastic reduction in the amount of powder that we would find within packaging. And that speaks to that gap in between the cap and the body. The evenness and smoothness of the HPMC film also contributes significantly to the higher quality performance of the thermo-gelation HPMC capsule in manufacture. So another notable difference in the ability for HPMC capsules to better tolerate heat, deviation, or hot storage conditions. And think of zone 3 or zone 4 stability regions when we think about that sort of thing. So our recommended storage conditions for gelatin capsules would be placed at 15 to 25 degrees Celsius. And deviations are certainly permitted with accompanied stability data. But as zone 3 and zone 4 actually call for 30-degree heat storage, this is somewhat out of the range and therefore more prone to instances of cross-linking as a result. So, or even heat damage of the capsules for that matter, for gelatin. HPMC capsules actually show a heat resistant well past that of gelatin, as you can see in the slide. So even up to 90 degrees, we're seeing very little change in the Vcaps Plus, the thermo-gelation HPMC capsule, as compared to the gelatin. So next we'll take a look at the dissolution characteristics of the thermo-gelation formed HPMC capsules. So when developing an alternative polymer, ideally you want a fast disintegration dissolution profile that's at least equivalent to the speed of gelatin, because gelatin at the end of the day is still the marker that everyone goes back to. It's been sort of predominantly used in the market. So in the inset of the graph, a smaller chart showing the disintegration rate of Vcaps Plus, the thermo-gelation capsule, shows that it's very similar but slightly higher compared to gelatin. Still very much within a normal relative standard deviation for disintegration of a capsule, and well below the requirement of 15 minutes. The effect is more pronounced, though, when you compare the dissolution rates of caffeine in both gelatin and the Vcaps Plus capsule. Here it's noted that the gelatin quickly releases the contents, allowing for dissolution to manifest rapidly, while the Vcaps Plus doesn't appear to allow dissolution until about 5 minutes into the dissolution testing. It turns out to be a phenomenon occurring in all commercially available HPMC capsules, either first or second generation. HPMC, being a drier capsule, a drier material compared to gelatin, needs a higher hydration period in order to pull water into the matrix and allow disintegration to begin. Gelatin's already got that 12 to 16% water content in it. It doesn't need that same hydration period, and that really accounts for the differences as when disintegration and dissolution take off. But when considering the use of HPMC capsules, it's really important to realize that not all the HPMC capsules are going to be an equivalent change from a gelatin capsule. So in that, HPMC capsules can be made by two very different manufacturing processes, one which uses a gelling system and one that does not. And the concern with the use of the gelling system is that inevitably some variability is going to be introduced. And this slide is very good at revealing just how much variability can be seen in vitro. So there's a lot of very good data here to draw from on this slide, and so I'm going to break it down just a little bit and go a little slower through this slide so that we can catch everything. So first, that across a range of pH and ionic strength content media is represented, and it can be seen you've got a variability in dissolution is very high, almost disconcertingly so. So as the differences in the media are further investigated, the implications of variability begin to really show up more fully. So in the graph, first consider the dissolution curves comparing pH 1.2 by the USP and pH 6.8 by the USP. At first glance, it would appear that the difference in pH is responsible for the amount of variability between the two curves. But if you'll notice, the 6.8 buffer system made according to the JP2 protocol gives a very similar dissolution to that of the pH 1.2 by USP. So pH really doesn't seem to be a significant contributor to the issues of variability that we see here. It turns out that the gelling system for this particular is carrageenan and does not tend to be as sensitive to pH. Rather, what is different between the pH 6.8 buffers made by either the USP or the JP2 protocol is that ionic strength between the two systems. That JP2 protocol was created to have a lower ionic strength just because of the contribution of the variability in dissolution that arises from secondary gelling systems. And this is further substantiated by noting the dissolution curves of pH 1.2 by the USP versus those of pH 1.2 in which potassium chloride has been added at either 2 or 9 gram quantities to increase the ionic strength of the buffers. And that in turn shows an increasing amount of variability between the three systems. So if variability is noted in vitro with ionic media, then there's an in vivo concern, and that should be just as suspect as well. Diets like the American breakfast, or those which fruit juice, vitamin consumption are all part of, would naturally then lead to variability in release rates for an HPMC that contain a gelling system because of the differing ionic strength of the actual meal that we're ingesting. Simulated milk fluid is routinely used to compare the ionic strength of a fed state against that of fasted protocol. So we'll look at that versus the pH 1.2 for fasted protocol. Again, a high variability exists between the two dissolution curves, and that's indicative of variability that can occur in vivo between fed and fasted states when an HPMC capsule that contains a gelling agent is used as well. So one essential point of that thermo-gelation, that Vcaps Plus capsule, is that it contains no gelling system. Because we're making these by a thermo-gelation process, no gelling systems whatsoever are needed, and without that gelling system afforded in a traditional manufacturing process, the level of variability is greatly reduced. So if we examine the exact same in vitro dissolution protocol that we examined in the previous slide with the thermo-gelation capsule, we see that the lack of gelling system greatly reduces in between the previous slide and this one. And I'll just go back a quick second and let you take a look at the variability again to have a better look at that. So no longer can a significant variability with a thermo-gelation HPMC capsule be seen from pH, ionic strength, or even fed and fasted states. But what you'll notice is one area that remains in question is that apparent lag time. So regardless of the ionic strength, regardless of the pH, it seems that this lag time of a couple minutes to 5 minutes is still prevalent. So despite this delay in the first 5 minutes of opening, though, when you take a look at the in vivo performance of Vcaps Plus, the HPMC capsule, it's very good. In a dose escalating experiment that was done, the median Tmax was observed at each case in 1 hour. So no drastic T lag shifts, nothing else. And that's indicative that the shell disintegrates quickly and allows the subsequent dissolution and absorption of the immediate release compound. So to further address any concern with that apparent lag time of the thermo-gelation HPMC capsule, and really better understand the performance of the capsule, we commissioned an in vivo study to compare the performance of the thermo-gelation HPMC capsule, the Vcaps Plus, compared with the gelatin ConiSnap product. Right before we get into that, though, I'm going to turn it over to Ernest for a quick poll question.
Thank you very much, Matt. It's now time for the poll question that we'll be running in today's session. If you could please select the answer that is relevant to you and then click Submit. So today's polling question and the selection of answers are: in your company, is the use of HPMC capsules non-existent, just beginning, readily used for some projects just as hard as gelatin capsules, used preferentially over hard gelatin capsules? Matt, what kind of answers are you expecting from the audience?
Actually, I think that we're gonna what we find internally is that the amount of people and research houses are embracing HPMC more and more for a lot of the physical characteristics that we talked about. And one that we really didn't touch on cross-linking tends to be a bear sometimes with gelatin. People don't find that to be the case with HPMC. And even that there are now commercial products that are in HPMC, people have less concern from a regulatory perspective. And we really do see it taking off in the R&D field.
Yeah. So audience, if you can just click on the answer which applies to you, and we will reveal the answer shortly. Okay, now we'll see the results. Very interesting results. Matt, would you like to give any feedback on those?
Not really. That's really not a surprising spread. So between beginning and readily used tend to be the bulk, and that is consistent with what we have seen. Some of the feedback from our customers have been, other folks when we're out in the field discussing with it. There are some that have not worked with HPMC at all. There's a lot of products being genericized, for instance, and if the product is initially in a gelatin, it tends to remain in a gelatin. So it really depends on the house. And I'm happy to see, though, you've got a 10% used preferentially over hard gelatin capsules, and that really speaks to what we're talking about, is that people have gained that confidence in the HPMC capsule and readily moving forward. They recognize the strength that the capsule can bring.
Thank you for your feedback. You may now resume your presentation.
Okay. So, as promised, what we're going to talk about is the bioequivalence study that was done between the thermo-gelation HPMC capsule and the hard gelatin capsule. So the study design that we have is listed here. So in this study, Excedrin extra strength caplets, we added them both the hard gelatin and the Vcaps Plus HPMC capsule. So we chose Excedrin extra strength because it contained 3 different biomarkers within it, each representative of a different BCS classification. The capsules were dosed according to the protocol that you see here. It's a randomized 2-way crossover study, and we took various PK parameters and collected them as part of the study. So in the results of from the acetaminophen marker, it's clearly shown that neither the difference in the polymer type nor the slight lag time in HPMC disintegration affected the performance of the product in the body at all. So here, gelatin's represented in the blue, and the HPMC Vcaps Plus is represented in the red on the graph. And it's seen that the performance is nearly identical with high confidence intervals between them as well. The same can be said for the acetylsalicylic acid marker as well. Again, the gelatin is seen in blue. Vcaps Plus HPMC is found in red on the graph. And when the values are examined in the data table below, the biggest difference was found to be the intra-subject variability, and otherwise the product performed very closely regardless of the polymer of the capsule, this time in a molecule from a completely separate BCS classification. And then we compare that to the third biomarker, caffeine. Even here, with a BCS class 1 molecule in which fast dissolution is characteristic, the roughly 5-minute lag time of the Vcaps Plus HPMC capsule shows no bearing on the overall in vivo performance between the 2 different polymers. Again, very similarly high confidence intervals are achieved. Okay, so with that, and also understanding some of the successes that we've seen with the 2nd generation HPMC capsule, interest, as we talked about, and I think was evident from the polling, interest in that polymer has truly escalated. So we began to look at a few applications of the polymer as well to increase their spectrum of use in pharmaceutical environments. So for customers who are working on pediatric and geriatric formulations to address the specific needs of those populations, or even augment existing pharmaceutical products, the HPMC capsule made by the thermo-gelation process is available in the sprinkle type format as well. So in this capsule, The opening force is roughly 5 times less than a traditional powder-fill Conisnap capsule, but still remains secure and consistent when you're thinking about filling and transportation of the pharmaceutical product. So these are available in both gelatin, but also now in that Vcaps Plus HPMC as well. Something else that we're working on currently for our colleagues in research and development who want to begin stability studies in HPMC early, really prior to final marketing approval, what marketing can say the color of the capsule and things like that are going to look at. We're currently developing an all-color HPMC equivalent to the gelatin that already exists, either available in a global color set or an FD&C color set, depending on the marketing needs. So we're developing an HPMC, a Vcaps Plus all-color capsule, to help those R&D colleagues in similar situations as well. And so hopefully with this, we've demonstrated that HPMC capsules can really broaden what we would consider working with the quality by design space for hard capsules, provided that the HPMC capsules are ones made and manufactured by a thermo-gelation, which contains no gelling system within the capsule. That lack of a gelling system really reduces variability in what's seen with traditional HPMC capsules. And then adding the in vivo data study to previously shown physicochemical benefits of the Vcaps Plus, it's really easy to see why these capsules are a great choice for the right first time formulation work. Really reduce trial and error efforts. So with that, I think I'm going to go ahead and turn it over to Mike for the next section of our webinar. So over to you, Mike.
Okay, thank you, Matt. So as Matt said as part of his presentation, we saw that the HPMC-based Vcaps Plus and gelatin capsules gave very similar PK profiles when we encapsulated the fairly soluble compounds from the Excedrin case study. And so when we have those types of compounds that are soluble and well absorbed, we saw equivalent performance. One might ask whether there are applications, though, for which the two capsule materials might give different PK performance. And it turns out there are such applications. And in particular, we can take advantage of the solubilization characteristics of HPMC for some of these applications. So that's what I'm going to talk about for the remainder of the time. So one mechanism that can lead to differences in performance results from favorable interactions between HPMC and poorly soluble drug compounds. It can reduce the crystallization rate of the drug that's supersaturated in the GI tract. So drug supersaturation can result from several different situations. For example, dosing a weekly basic API or dissolution to the ionized drug form in the low pH environment of the gastric leads to a significant supersaturation upon transit of the drug to the higher pH of the small intestine. Supersaturation can also occur when using a high-energy drug form such as a high-energy salt, a solid amorphous dispersion, or a lipid-based solution, which can provide dissolved drug levels greater than the crystalline solubility. So in all these cases, HPMC-based capsules can potentially maintain the resulting supersaturation by inhibiting crystallization of the drug. And by having the HPMC in the capsule shell, this creates more room inside the capsule for increasing the drug payload, for example. So today I'll touch on drug sustainment from all the formulation types I just mentioned, that is high energy salts, amorphous forms, lipid formulations, and weekly basic drugs. I'll start with the weekly basic drugs. In the next couple of slides, I'll show examples of dissolution profiles of several weekly basic drugs in a simulated gastric fluid to simulated intestinal fluid transfer test, where we assess sustainment of supersaturation upon transfer to the high pH of the simulated intestinal fluid, where the weak bases convert from the highly soluble ionized form back to their low solubility free base form. In these studies, we compare the performance of weak bases in both Vcaps Plus and in gelatin capsules. And we would expect the sustainment of the drug to depend on the drug concentration relative to the gastric and intestinal solubilities, on the tendency of the API to crystallize, and also on the type of capsule, of course. And as you'll see from the data, all 4 of the drugs that we tested here showed improvements in sustainment using the HPMC-based capsules relative to gelatin. So these are the 4 weekly basic drug compounds we used, and you can see that they're all relatively soluble in the ionized state in the low pH typical of the gastric environment, but have much lower solubility at the neutral intestinal pH, which can lead to highly supersaturated states of the drug in the intestinal environment in vivo. And here we show results from all 4 of the different compounds. For each of the plots, the green curve is from the drug encapsulated in gelatin capsules, and the blue curve is from drug in HPMC-based Vcaps Plus capsules. And from 0 to 30 minutes on the x-axis, the formulation is in the simulated gastric fluid at pH 2, and then at the 30-minute time point, it's transferred to the simulated intestinal fluid at pH 6.5. And note that there's a 2-fold dilution upon transfer from the gastric to the intestinal fluid. So the initial concentration drop to half at 30 minutes is from that dilution. But you can see from the profiles that the drug sustains much better in the SIF using the HPMC-based capsules versus the gelatin. So it's a fairly broad behavior here. So moving from the weekly basic crystalline APIs to amorphous forms, such as solid amorphous dispersions, including spray-dried dispersions, just wanted to note that such solid dispersions are used extensively these days to solubilize poorly water-soluble compounds for oral delivery. And these solid dispersions are often comprised of a drug dispersed in a polymer matrix. And there are quite a few requirements as shown on this slide around the components of the solid dispersion with regard to manufacturability, dissolution performance, and physical and chemical stability. And sometimes it can be difficult to achieve all these objectives using a single or even multiple matrix materials in the solid dispersion, particularly when we want to achieve high drug loading in the dosage form. So for some drugs in solid dispersions, HPMC can help sustain supersaturated drug levels by interacting favorably with the dissolved drug to prevent crystallization. And instead of being put into the dispersion itself, the HPMC can be incorporated into the capsule shell as it is for Vcaps Plus. And this takes some constraints off the design of the solid dispersion, allowing the formulator to choose the solid dispersion matrix to achieve rapid dissolution or physical or chemical stability, for example, rather than having to choose the matrix polymer to achieve all the functions shown on the slide here. So this slide shows a few of the material properties that are typically needed for a solid dispersion, such as the spray-dry dispersion. And so it's often advantageous to have a high glass transition temperature for good physical stability. Also, dispersion would typically have some hydrophilic components to achieve rapid dissolution, and then also some amphiphilicity at the end there to have some interaction between the poorly soluble drug compounds and the polymer to inhibit crystallization. So it's this last function that can be provided, at least in some cases, by the HPMC in the capsule shell to allow the formulator to focus on the first 2 functionalities in the dispersion itself. So it can be difficult to predict a priori which drug compounds will interact with HPMC or any other kind of polymer to provide good sustainment. And one way to quickly assess this is through a relatively rapid in vitro screening test, as shown here. The poorly water-soluble drug is dissolved in an organic solvent, and then supersaturation is introduced into the test through a solvent shift into a simulated gastric fluid or simulated intestinal fluid containing pre-dissolved or pre-suspended polymer. And the sustainment of the resulting supersaturated drug levels is monitored via a UV probe. A typical result of this screening test is shown here, where one polymer, polymer 2 shown in green that is, was found to inhibit crystallization of the drug relative to no polymer shown in blue, while another polymer designated polymer 1 here in black, did not help sustain drug supersaturation. In fact, may have promoted crystallization. So that screening test can be used with a variety of polymers, including HPMC, to see if capsules made of HPMC, for example, may sustain supersaturation of a drug in an amorphous form. This screening can be done using not only different polymers, but by varying the concentrations of drug in polymer to assess the capacity to sustain dissolved drug levels for compounds of interest. So here we show the results of varying the drug concentration. On the left is a plot showing sustainment of drug with various concentrations without HPMC added. And you can see that the precipitation to the crystalline form is rapid at concentrations above 25 micrograms per mil. The right-hand plot shows similar data with the addition of 0.1 mg per mil of HPMC, which shows maintenance of much higher drug levels that can be sustained as a function of those higher levels. And the model compound here is Erlotinib. So moving on then, one can proceed to test the actual solid dispersion of the drug with pre-dissolved HPMC to assess any improvement in sustainment as a function of drug or HPMC concentration. Here we show an example for an Erlotinib SDD now, made using Oidrogit L100, which is an enteric methacrylate, as the SDD matrix polymer. And you can see from the blue curve that Oidrogit L100 does not sustain Erlotinib well by itself, but with the addition of HPMC in the media, sustainment of the drug levels is better. And notice that although the performance improves slightly with increasing HPMC concentrations, that actually very low HPMC concentrations work very well, suggesting that at least in some cases, we may not need to use a lot of HPMC to have a positive impact on solubilization. So based on the previous slide, we'd expect HPMC-based capsules to help solubilize the Erlotinib SDD that we are showing here relative to the gelatin capsule, and that is indeed what we see in this dissolution test now, where we've encapsulated that SDD both in HPMC-based Vcaps Plus capsules and in gelatin capsules. And you can see the much better sustainment from the HPMC-based capsules in this test. So all that I've shown you so far looks pretty positive in vitro, and the question is, does this provide a benefit in vivo? And that is, can the sustainment of supersaturated drug levels improve absorption? And so here on this slide are shown two examples from the published literature where it did indeed improve oral exposure. So on the left-hand part of the slide are results from a clinical study of Dabrafenib, in which the high-energy mesylate salt of the drug was encapsulated in both HPMC and gelatin capsules, and the AUC was significantly higher for the drug encapsulated in HPMC, as you can see. And the plot on the lower left part of the slide shows in vitro data collected by the authors of the study that showed improved sustainment of solubilized drug levels for the HPMC-based capsule, which they suggested was responsible for the improved exposure in vivo. And then on the right-hand part of this slide is a different study. This is a dog study of a different type of high-energy form, namely a SED formulation of a poorly water-soluble drug, showing again higher exposure from formulations encapsulated in HPMC versus in gelatin. If we compare the open triangles for the HPMC-based capsules to the dark-filled circles for the gelatin capsules. And likewise, in the lower right plot is the in vitro dissolution data that goes along with this, showing the positive impact of the HPMC on sustainment of the drug in vitro. So we've seen through a couple of examples that HPMC-based capsules can sustain supersaturated drug concentrations, and that this can translate to improved oral exposure, at least in some cases. So we might ask, what is the range of drugs or formulations for which the use of HPMC in this way might be advantageous? And it turns out that a large fraction of drug molecules in development are either weakly basic or have poor water solubility and are therefore likely to be formulated as high-energy supersaturating drug forms. So we've looked at our own portfolio of molecules that we've formulated at Capsugel and determined that from a sampling of 500 or so of our recent molecules, that 40% of them are weakly basic. that is having a basic pKa between 2 and 7. And almost all of these are poorly water soluble, as shown in the plot on this slide, which displays solubility and FASF on the y-axis versus basic pKa on the x-axis. So that's a significant fraction of our development portfolio. The applicability space extends well beyond weekly basic drugs, as non-basic poorly water-soluble compounds are often formulated as high-energy forms, such as amorphous dispersions. They can supersaturate and potentially benefit from crystallization inhibition from HPMC-based capsules. So shown here is the well-known developability classification system, with some estimates suggesting that a majority of drug compounds in development, as high as 70, 80, or even 90% in some portfolios and therapeutic areas, have poor water solubility. So these are the ones that would likely be formulated in a high-energy form that may benefit from crystallization inhibiting polymeric materials. So this suggests that the use of sustaining polymers such as HPMC in the capsule shell could potentially have a positive impact on quite a few drug development programs. So with that, I'll wrap up the presentation by just summarizing the last section. For some drug compounds, HPMC capsules can inhibit drug crystallization from a supersaturated state, and particularly for applications requiring solubilization, such as compounds that are weakly basic or that are formulated in amorphous form or another high-energy form. And of course, we're actively engaged here at Capsugel in research focused on understanding and predicting the relative performance of both the gelatin and HPMC-based capsules, and we're glad to engage with clients on their specific applications whenever helpful. So with that, I'll end the presentation and thank our audience for participating today, and we'd be glad to answer any questions in the few minutes that remain. Thank you.
Thank you very much, Michael. We received a wide range of questions from the audience. I'll get straight into those. We have a question here. Are the HPMC capsules available in DB sizes, i.e. suitable for over encapsulation in clinical trials?
This is Matt. I'll take that one. So we do have some selective DB sizes available. Typically, these are the larger sizes, and these are perfectly suitable for over-encapsulation work. I think the one caveat would be if you've got a drug that is known to be very fast release, and you need to take very early time points at the 5 or 10 minute periods and be equivalent to gelatin, you should be aware because of the slight dissolution and or slight difference in lag time, you may have a different result. So it will warrant some further investigation on things where you know you have to take a very early time point. Usually the difference that you see in dissolution times are equivalent by the 20-25 minute range, but for early time points, definitely going to need some further investigation.
Thank you very much for your detailed answer. Another question here. Any data on accelerated conditions, 45-75, or their effect on dissolution rates?
As far as HPMC capsules go, you're not going to find any difference that we've seen in traditional room temperature stability work versus accelerated stability work. I do see a difference, or you will see a difference occasionally in gelatin, but I don't see the same differences pop up in HPMC. And that's just because the cross-linking that is occasionally shows up in gelatin because of heat and humidity conditions that are outside of recommended storage conditions. They simply chemically don't exist in HPMC the same way. And so we don't see the same slowdown that you would typically prompt a tier 2 testing in an HPMC capsule that's got thermo-gelation. Again, the caveat is that we're looking at the second generation capsules that don't have any gelling system in there, because that will provide some variability, not necessarily because of the stability, but just because of the gelling systems that are in there.
Thank you for your answer. If I can remind the audience, if you'd like to send in a question, just remember to type it in the Q&A box. The next question that we have here is, which sizes are available for sprinkle caps?
Oh, that's a really good question, and I want to, don't quote me on this. We have the larger sizes available at the moment and not the smaller, but I believe we have a 0 and a 00. Feel free to contact me. I think that my email address is listed or my contact information is listed at the end of the webinar. Please feel free to contact me, and I'll be able to confirm that for you.
Great. We have a question here, especially directed at Matt. What are examples of products that are currently encapsulated with Vcaps Plus?
Okay, so you can really think about encapsulating an entire range of products. The world is your oyster when you think about HPMC. I'd say the list of products or molecules that you couldn't put into an HPMC, I could list easier than the amounts that you could. So I think HPMC is really suitable for most products that are out there, other than the really oxidation-sensitive type products. And we find a particular draw for people that are doing hygroscopic or moisture-sensitive type work, because we can have the lower water levels and lower amount of interaction between an HPMC and formulations that are out there. So we see a particular draw to that. And then, of course, anybody that's challenged with aldehydes, formaldehydes, and similar type cross-linking agents as part of their formulation, they're going to find those to be super beneficial within an HPMC compared to a gelatin as well.
Thank you for your answer there, Matt. Very interesting question here. Are there any mechanical or capsule filling machine differences between HPMC and gelatin?
Okay, that's a good question. So we have investigated that both in-house and externally at customers. So we'll have a technical service engineer or team work with customers that are using HPMC capsules for the first time in case they have any questions. And really, in essence, what we have found is that when we've done the studies, you get about a 99.9% yield rate using an HPMC capsule compared to gelatin. So in effect, no difference at all in running an HPMC versus the gelatin. It is going to require some tweaks as far as the equipment goes. Nothing that I think would be a capital expenditure, just changing things like vacuum settings, closing settings, the amount of force required to do that, just simply because HPMC is a softer material overall than gelatin is.
Thank you for your detailed answer. We have a question here directed at Mike. Is the amount of HPMC in a capsule shell enough to sustain most low solubility drug?
Yeah, that's a good question. So it depends obviously on the drug compound itself, how much HPMC is needed to help sustain supersaturated drug levels. But we saw, for example, in the case of Erlotinib, that very little was needed, on the order of 0.1 mg per mil. So if you think that there's approximately 100 milligrams of HPMC in a capsule, that gets to be, at least a large fraction of 1 milligram per mil in a youth environment. So there's a substantial amount of HPMC around that can be used to sustain drug compounds. So for a large range of compounds, there probably is enough in the capsule shell to do so. Thanks.
Thank you very much for your answer. We've received a lot of questions, so keep those questions coming in. The next question that we've received is, it was mentioned that the HPMC capsule can help sustain weekly basic drug? Can it be used to sustain weakly acidic drugs?
Yeah, thanks for that question. Obviously, basic drugs are a little bit more common than acidic drugs, and basic drugs also have the issue that they can supersaturate due to the low pH of the gastric, and then so they tend to be more likely to be supersaturated when they are then transferred to the simulated intestinal fluid or the actual intestinal fluid where the pH is higher, and so they tend to be more likely to supersaturate even in the crystalline form due to the pH difference between the stomach and the intestine. Acidic drugs, on the contrary, have a low solubility in the lower pH gastric environment and a higher solubility in the simulated intestinal fluid or in the actual intestine. So they're less likely as crystalline forms to supersaturate and to need this type of supersaturating excipient. On the other hand, if weakly acidic drugs are formulated in a high-energy form, a high-energy salt, or an amorphous form, then they too can supersaturate and benefit potentially from this type of crystallization inhibiting excipient. So good question, and indeed, in some cases, it may be applicable to acidic drugs as well as weak bases.
Another question here from the audience. On a percentage basis, what is the supply of gelatin relative to HPMC? Plus, the same question for new releases.
I'm not sure that I know the answer to that right off the top of my head, but we'll certainly investigate it. We've seen, I would say, over the past 5 years, a large uptake in the amount of usage of HPMC, both in pharmaceutical and also in the health and nutrition. Health and nutrition by far has embraced the HPMC capsule, being a non-animal type product. And moving forward, we see many more sales in HPMC in the pharmaceutical world as well over the past few years. But I don't know if I could give you a percentage off the top of my head today on between the two. We'll investigate it, and I'm happy to follow up with you on it.
Great, thank you very much. Another question here. For a fecal microbiota transplant, so FMT study, it is necessary to use double-enteric caps that will only release the product in the intestines. Which version of Vcaps would be best for this application?
Okay, that's a good question. I actually, if you're looking for an intestinal release, I would not recommend either the first or the second generation HPMC capsule. The Vcaps Plus, the thermo-gelation that we talked about, is really an immediate release product. It is designed to open in the gastric environment and release right away. The Vcap or any HPMC that's got a gelling system is going to have a longer time opening in the low pH, but it's still going to open in the stomach. So I think you are better suited for products like this positioning towards an enteric capsule. We have developed an HPMC enteric capsule called the Vcaps enteric, of all things, that's designed just for products like this, and I'm happy to speak about the performance of that with you directly. And I think we've got your contact information, so yeah, I can reach out to you or vice versa regarding this type of product. But in answer to your question, at the end of the day, I don't recommend either. They're both going to open in the stomach. One, albeit one later than the other, but it's not going to give you the delivery that you're looking for.
Thank you for all your questions, but unfortunately we have run out of time for any more questions. So that just leaves me to thank Matt and Michael for what was a great presentation, and thanks to Capsugel for sponsoring this session. To the attendees, you will receive an email tomorrow telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. Please continue to ask questions, vote in the poll, and interact with the widgets as the on-demand version remains interactive. We look forward to sharing further webinars with you, so please keep an eye out on our website and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. Thank you once again, and I all hope you have a great day.